This is the lecture for Lab 5 on cells and microscopes. All living things are composed of cells. So therefore, cells are considered the basic unit of life. All cells must have a membrane to separate the inside of the cell from the environment. They also must have DNA, the molecule of information to determine what the cell can produce and what it can do. And the last thing that all cells must have are ribosomes. Ribosomes are responsible for producing proteins, which form the enzymes and catalyze biochemical reactions. There are two types of cells found on Earth, the prokaryotes and the eukaryotes. And first I will go through the prokaryotes. There are two different subtypes of prokaryotes, the archaea and the bacteria. Prokaryotic cells are very, very small. They are between 2 and 8 micrometers in length. Prokaryotes are so small that they do not have organelles, but they do have a nucleoid, which is the area where you find the genetic material, the DNA, and they also have a cell wall. This is a diagram of a typical prokaryotic cell, specifically a bacterial cell, and here you can see that it has a membrane. It has DNA, which is found in the nucleoid. That's the area where you find the DNA. It also has ribosomes. And like most prokaryotic cells, it has a cell wall and it has some other structures. The other types of cells you find are eukaryotic cells, and they belong to the domain of eukarya, which includes the protists, the animals, the plants, and the fungi. So pretty much everything besides the bacteria and the archaea. Eukaryotic cells are much larger than prokaryotic cells. They're on the order of 10 to 100 micrometers in length. They are also much more complex than prokaryotic cells, and they have organelles. Organelles are defined as membrane-bound areas of cytoplasm that have a specific shape and function. And most eukaryotic cells have cell walls. The plant cells, the fungal cells, many of the protist cells have cell walls. The only exception are the animal cells. Animal cells do not have cell walls. As I mentioned before, eukaryotic cells are very complex and they have organelles. And again, the definition of an organelle is a membrane-bound area of cytoplasm that has a specific shape and function. The first organelle I want to talk about is the nucleus. The nucleus is the organelle that contains and protects the DNA. Also, the nucleus has a double membrane. So it's composed of two membranes called the envelope. And usually the nucleus is the largest organelle in the cell, and it's the one that is easiest to see. Another structure that you will see in eukaryotic cells is the nucleolus. And the nucleolus is inside the nucleus. So if this is the nucleolus is this darker staining area inside the nucleus. The nucleolus is the site of ribosome subunit synthesis. So this is the area in the cell where the different subunits of the ribosomes are produced. And then those subunits will leave the nucleus through the pores. So they will leave the nucleus to go into the cytoplasm using the pores for exit. The next organelle I want to talk about is the endoplasmic reticulum, otherwise known as the ER. And the endoplasmic reticulum extends from the nuclear envelope. So here, this purple or pink colored nucleus, this is the nucleus, and the membrane extends out from the nucleus and becomes this blue ER. There are two types of ER, the rough ER and the smooth ER. The rough ER has ribosomes bound to it, so it looks bumpy. That's why they call it the rough ER. And this is where protein synthesis and modification occur, because ribosomes produce the proteins, and those ribosomes are found on the rough ER. The other type of ER is called the smooth ER, and it's called the smooth ER because it does not have ribosomes bound to it. Instead, it has a lot of enzymes embedded in it, which are responsible for lipid synthesis, making lipids. And some of those enzymes are also involved in drug detoxification. The Golgi apparatus is another organelle which is found close to the rough and smooth ER. The purpose of the Golgi apparatus is to receive and modify the proteins produced from the rough ER. So the Golgi apparatus will take those proteins, modify them so that they are functional, and package them into vesicles. So because of this, it's often referred to as the packaging and distribution center of the cell. So it would receive products from the ER here, process the products in the Golgi apparatus, and then package them into vesicles and deliver them to another part of the cell. The mitochondria is another organelle that is found in the cell. The mitochondria is often referred to as the powerhouse of the cell because its purpose is energy production, and that energy is in the form of ATP. The mitochondria is surrounded by a double membrane, an outer membrane and an inner membrane. The inner membrane has many folds in it called cristae. Those folds increase the surface area, and that is important because that increases the amount of ATP that is produced. The mitochondria contain their own DNA. They have a small circular piece of DNA, and they also contain their own ribosomes. There are a couple miscellaneous organelles I would like to go over. First are the lysosomes, and lysosomes are vesicles, little membrane-bound areas that contain digestive enzymes. So their purpose is to digest food and waste. So an analogy is to the stomach for the human body. These can also be referred to as suicide sacs because if the membrane ruptures and those digestive enzymes are released inside the cell, they can digest the important organelles for the cell, which will result in death. Another organelle are the peroxisomes. Peroxisomes are also vesicles and they contain catalase enzymes, and those catalase enzymes are used to break down lipids. The last structure that I want you to know about are found in animal cells only, and those are the centrioles. Centrioles are found in pairs, and again, found only in animal cells, and they are composed of microtubules. And their purpose is to organize the scaffold, the fibers that are used during cell division to pull the chromosomes apart from each other. This is a diagram of a typical animal cell, and you can tell it is an animal cell because there is no cell wall. And the organelles I have already talked about, here is the nucleus, usually the largest, most visible organelle that you can see. Here's the nucleolus inside the nucleus. Then you have the rough ER here and the smooth ER. Difference between the rough ER and smooth ER. Rough ER has ribosomes, so it produces enzymes. Smooth ER does not have ribosomes. It produces lipids and detoxifies the cell. Then this is the Golgi apparatus here that receives the products from the ER, packages them, modifies them, and delivers them to different parts of the cell. Then here's a mitochondrion, produces ATP for the cell, and the centrioles and lysosome are also pictured here. Plants are composed of eukaryotic cells also, and there are a couple of structures that are unique to the plant cells. The first structure are the vacuoles, and vacuoles are a big storage area and also used for water balance. Chloroplasts are the other structure that is unique to plant cells, and they perform photosynthesis. So you only find those in cells that perform photosynthesis, which are most of the plants and many of the protists. A chloroplast is surrounded by a double membrane, and it also contains its own small circular piece of DNA and its own ribosomes. This is a diagram of a plant cell, and this is definitely a plant cell because it has a cell wall, so surrounded by a cell wall. It also has a nucleus with a nucleolus. It has the rough ER, it has the smooth ER, it has the Golgi apparatus, which is receiving products from the rough ER, modifying them, packaging them, and delivering them. It also has mitochondria, and in addition, the special plant structures. It has the central vacuole, which looks like this big empty area, that's for water balance, and it also has the chloroplast. So the chloroplast here is the organelle responsible for photosynthesis. In lab, you will be using a microscope to look at samples of different cells, and it's important that you know the basic parts of the microscope. So the first part of the microscope is the stage. This is the stage, and that's where you put the slide. You put the slide in the stage clip on top of the stage, and that will hold it in place. Then there are two sets of lenses. Up here is the ocular lens. It's called ocular because it's the one you look through with your eye, and it magnifies 10 times on its own. The other set of lenses are on the nose piece at the end, and they are called the objectives because they will be close to the object you're looking at. And you will have 3 different levels of objective lenses. You'll have the low power, which is sometimes called scanning, and it magnifies 4 times. Then you will have a medium power that magnifies 10 times, and a high power objective that magnifies 40 times. When figuring out total magnification, you take the ocular magnification and you multiply it by the objective magnification. So the low power is 4 times. If you are looking at the sample using the low power objective, then your total magnification is 10 times 4, so it would equal 40 times is your total magnification. When focusing on the slide, you are going to use 2 knobs that are found on the side of the slide. The first is the coarse focus. The coarse focus knob raises and lowers the stage for focusing. Once you find the cells in focus using the coarse focus, then you switch to the fine focus knob, which sharpens the image, and it will not move the stage. In class, you will look at a slide that has mixed bacteria on it. So these are the prokaryotic cells, and the bacterial cells are very, very small, so you won't be able to see too much detail on them, but you should be able to see some cells that look like rod-shaped cells, and maybe possibly smaller rod-shaped cells or circular cells. Another slide that you will observe is of epithelium. Epithelium is a tissue, and a tissue is made up of many different cells. And epithelial tissue is usually like a covering, like skin. And in this, you have to be sure that you can identify individual cells. These empty spaces here are actually cross sections of blood vessels, so those are empty areas. When you are looking to identify the cell, you want to look at the cells that are surrounding those empty spaces. So actually, this is one little cell there. So you want to get used to identifying the cells. You should be able to see the cell membrane, and then the nucleus is that brighter pink round structure inside the cell. You will also look at some cheek cells, and cheek cells can be a lot easier to see. And for the cheek cell, this would be one individual cheek cell here, and that is the outside of the cell membrane, and of course the organelle that is easiest to identify is the nucleus. Another sample that you will observe are live protozoa. So protozoa are a type of eukaryotic cells, so they're much larger, they'll be easier to see, and these are alive, so they will be swimming around in a water solution. So you will be able to see them and many different types of protozoa in the sample. I have included a link to a protozoa video, so if you are curious to see what different types of protozoa look like when they are moving, you can click on this link and watch the video. And some of the same protozoa are the ones you will see in lab. You will also observe some plant cells in the form of onion cells, and for this you should be able to identify the cell wall, which will be the nice rigid structure that looks sort of like bricks, and again you should be able to see the nuclei, the nucleus, because that's the most visible organelle. The last slide you will look at is of a lichen. And a lichen is something that you have actually probably seen, especially on trees. It looks like a mold that is growing on the bark of trees. Sometimes it's gray or bluish or green, and that is a lichen. It's an organism that is a combination of a fungus and algae. And you will look at a slide microscopic view of a lichen, and you should be able to see some of the algal cells. Those are photosynthetic, and they produce glucose by photosynthesis to help provide food and energy for the fungal cells, so for the mold cells. So a lichen, actually, it looks like it may be damaging the tree, but it's not, because the fungus actually gets energy from the algal cells. So it's a symbiotic relationship.